So in this presentation, we're just going to quickly go through. So we've talked about some of the common file types. So we talked about FAST5 and FASTQ because that's what we've come across so far. But there are other file types that you might come across when you process your data a bit more. And then we're also going to talk about how to use TB Profiler and how to look at and analyze the data in that. So I think this slide we've already been through. So John showed you the left-hand side, and what we're going to go through in this presentation is the right-hand side. We're not actually going to see all of this happen because TB Profiler does this in the background. So either you will upload your data to their website, or you will download the software onto your computer, but it just runs it in the background. But there are different programs within TB Profiler that do each of those different steps. And then what it does is outputs a file that you can read yourself. Oh, there you go. So it's saying Minimap does that. It's gone. Right. So you've got your reads. You're happy with your reads. You're happy that the quality of your reads is good. So what you then might want to do is put your reads together to form the genome. So there's two different ways of doing this. There's something called de novo assembly, which is where essentially like putting a jigsaw puzzle into place, but they're all blank. So you don't have any reference, you can't see any of the blue corners, you don't know where any of the bits are, and what the programs do. So a de novo one for, what's the de novo one? Fly is the de novo assembly for MinION. So what it does is essentially it goes along, looks at all of your reads and tries to find out by itself where they all fit together. The reason you might do this is because you might not know what species it is. It might even be a species not known to science potentially. So it's kind of, it will bring up what are called contigs. So contigs are the program's best attempt at putting these bits together. You might find that maybe it can't quite work out in certain areas. You might get one whole contig, which would be a whole genome. You might get 2 or 3. It's just its best attempt to put all the pieces into the right place. The other way of doing it is using a reference-based assembly. So that is using Minimap. So this is what TB Profiler uses. And what that does is you have specifically put in a reference genome. So it uses H37RV. So you've already got a baseline and it kind of knows where to put things. So it's a bit like having your jigsaw puzzle the right way around because you can see roughly where you go and you've got your picture in the first place and you put them into the places where they should fit. So generally reference assembly is more accurate because you've already got something to align it to anyway. But you can use either of these options. As I say, we've looked at FAST5, we've looked at FASTQs. There's also something called a SAM file and a BAM file and a FASTA file and a VCF file. So SAMs or BAM files, these are the files that you get once you put all your sequences together. So what they are is, if you remember when we showed you, so Keith showed you and I also showed you, where you get those piles, stacks of like coins or pancakes or whatever you want to call them, against the reference genome in Artemis. So that is essentially your SAM file, or at least it's actually your BAM file. You just convert it into a readable format. And it's not the individual sequence, it's all of your reads basically piled up where the computer thinks they should go. So yeah, the SAM file is the output file, and then you can just change it so it becomes a BAM file, and that becomes a compressed version of that, and it's much more readable. With the BAM files, you also have to generally give the programs that use them an index file. So you have to sort it and then you have to index them. And what that does with the SAM and just a basic BAM is all it is a folder with bits of reads in some kind of order. They're not in any real order. What you do when you sort them is you put them in the right order. So sorting basically says, this looks to be at the front of the genome. This bit might be next to it, and then this bit. And then when you index it, that gives each of those bits that number. And so if you put it in Artemis as a BAM file, it would have no idea where each of those sections went on that genome. So all of these sort of processing bits are just helping the programs that you're analysing it with to actually understand what you've got. Yeah, so that's exactly what I was saying. So down here, this is kind of the equivalent of the reference genome. And if I had zoomed out a bit, you'll see what the actual sequence is for H37RB. So it might be AT, GGGGG, whatever. These are our reads that have been aligned by Minimap. And it knows that, as I say, this read goes here because it aligns with this, and then this read goes sort of next door to it. And they've all been given a number so that the computer then knows this read, that clearly goes there. So that's basically what that is. What you need to do, though, if you want one single sort of read or one single base for each of your sections, is to then convert it into a FASTA file. So like John was saying earlier. So what that does is it kind of compresses them all down. And this is where depth is really important. When you change it from a BAM file to a FASTA file, what the computer does is it takes every read at each point and says, right, okay, we've got 50 of these, 90% of them are A, 10% of them are G. So let's say that's probably going to be an A. So this is why having a really big depth is really important, because if you've got 10 of them and 50% are A, 40% of them are T, and then the other rest is sort of C or whatever, then it's going to be less confident that what it thinks it's got is actually what it's got. If you have 100 and 99% are A, then that's going to be very confident that that base is A. But it does its best. And what it brings up is basically just one long read of what it thinks each of those bases are. So it's essentially compressed that BAM file into its best guess for whatever each of those bases is. So as John said with the FASTQ file is just the quality version of the individual reads, and this is the FASTA file of our whole genome now. It's much more simple. It just has a header and then it just has the sequence. So there's not too much to really explain about this. Yeah, just 2 lines. A VCF file. So if you are then wanting to look for variants, so you've got your reference genome, you know the sequence of that, you now have your FASTA file and you have the sequence of your isolate or whatever it is. To identify whether they are exactly the same all the way along, or there are SNPs, or maybe there are indels, or whatever it is you're looking for, you can edit this into a VCF or variant calling format file. And there are various tools that you can use to do that. VCF tools does it. Can't remember off the top of my head any of the other ones, but there are lots of different programs. Claire 3 is another one, I think. And Medaka does it as well. There are lots of them. And what those programs do is basically they compare your reference genome with the genome of the organism you want, and it goes along and it looks, it says, okay, they're both A, they're both T, they're both A. This one's G, this one's C. There's a SNP there. It can do the whole genome, although that might take some time because that's quite a lot of information. Or you can specify, I want you to check in, say, RPOB. So you have to find the base numbers for RPOB, so start to finish, and say, I want you to check specifically between base 23 and base 83, and it will check within those sections. Otherwise, you get enormous files with a lot of SNPs, unless it's TB, and then actually it probably won't be so many SNPs. You can view VCF files in genome viewers or in Notepad, and I think the next slide will show you that. And it is designed for very large scale data storage. So the Notepad file is going to be, it's only a snippet of it, but they can be massive. And as I say, you can set them to filter it so you see only certain sections, or only one single SNPs, or specifically insertions, or you can change all sorts of things. So the programs are quite powerful, but they do also need quite a lot of computer energy. So this is the top of a VCF file that I have just copied and pasted. It's got lots of information at the top that explains, so this goes on to the right quite a long way. It's got lots of different scores. You can see SGB, VBD, and it says at the top what each of these are. But the thing you're probably most interested in is the sort of left-hand side. So you can see, so what's a bit annoying is that this is chromosome belongs to this section, that pause belongs to the second line. So what you're actually interested in is the first line where it says GG, and then the second line that says TC, And that's reference genome, so that's GG, and then alternative, which will be your isolate, so that is T and C. So it's gone along through the whole genome, and it's saying, right, the first SNP that I have found is at position 6140, and the original one, so in H37RB, that's a G, but in the alternative one, that is now a T. And so all it does is it just goes down all the way. Oh, I've explained it here. So yeah, so chromosome, oh, we're going the wrong way. So yeah, the position is where it is on the chromosome, and that obviously depends on whether you've done the entire chromosome or just a small bit. The reference genome is there, and then, as I say, the alternative is there. Why have I done this? I can't even read my book. I think I'm just pointing out that there's lots of different sort of things that it specifies. So these files can be quite complicated. And it's something that you might want to have a look into before you start doing it to actually identify, because it gives you information about the quality and how confident it is about its SNP call. So it's got the Fred scale scores. But yes, I think rather than try and explain all the different bits, I think it's probably better to just kind of gloss over that for now. Okay, the TB Profiler is a program that has been developed, I think it's developed at LSHDM by somebody called Jodie Falan. It can detect drug resistance comparing mutations, predict phenotypes, and also it can look into lineages as well. There are two different versions, as we said. So there's a browser-based one, and then there's the command line one. But for the command line one, and that's the reason why we didn't decide to do this, you need either Linux or Mac for it. So if you have a Windows computer, you can use the Windows subsystem for Linux, which I do sometimes. But actually, because the internet at UCL where I work is very powerful, it's much quicker for me to upload it to the internet than it is to run it on my fairly slow computer and wait for it to do it, you know, using the command line. But you can use either of them. The pipeline that they've developed is evolving rapidly and has done sort of the whole of its lifespan. And currently, although hopefully that is still the case, the version is 4.4.1. The reason it's developing all the time is because, as we were saying earlier, they keep finding new SNPs for all of the different drug resistances. So they're trying to keep up with that. And it does sort of give you a list of all of the drug resistances and the SNPs that it's actually using. It works with Nanopore data and also Illumina data, and I think it works with other platforms too. The pipelines are generally standardized, so it does the same thing for all of the different types of data, but it has different programs. So WA is the program that we'd use to align Illumina data, and then it uses Minimap to align Nanopore data, because obviously those programs only work with those types of data, but it's essentially outputs exactly the same thing. Yes, so I've already said that they update it based on what's coming out in the literature. This is just a slightly complicated version of what actually happens. So what they do is they can guppy for you. So what they use, so you input your FASTQ file, it then trims off the barcodes, and then it uses Minimap to align it to H37RB. And then once it's got its BAM file, it then does all sorts of different things to give you the output files that you need. There are pros and cons to the different versions. I like the web-based one. It gives, I think, a much nicer output, a more easily understandable and readable output file. It's very simple to use. You literally just upload it and it gives you a number, and then you put the number in when it's done, and it brings up your report. It's very user-friendly. And it's very easy to interpret. The con is it's limited on file size to 4 GB, which is still quite a lot. It used to be 1 GB, and that actually was quite small. So actually the only files you could upload probably weren't large enough or wouldn't have enough depth to really actually give you the full response. But now 4 GB is quite good. But it does also require a stable internet connection. And if you're uploading 4 GB of data for one file, then that's obviously going to take a bit of time. You could then use the command line if your internet is a bit dodgy. Once it's downloaded, it doesn't require the internet, although you would need to keep checking to make sure you've got the most up-to-date version. It can be fast, although that does depend on your computer, and it's unlimited in file size. And you can also, actually on both of them, you can batch, so you can add multiple files as you go along. As I said, it only works on Linux and Mac, Linux and Mac. You can't use it on Windows, but you can use the Windows subsystem. You must have a fairly basic understanding of line command, and actually, as we found, it's not the actual running of it, it's the installing of the program and making sure it actually works. I mean, when I installed it, I had a bit of issue actually installing it. But once you run it and once you've got the command, then actually it's relatively simple. And I've also found that the output, so it automatically outputs to a JSON file, which you can read on something like Firefox, but you can also request that it outputs as a text format, which is a bit easier to read. We can have a look at both of those. So that's the website link, and this is kind of the website. We can have a look, I think, in a sec. But basically what you do, you add your sample, so it says upload, and then it says, do you want to add paired or do you want to just add one? So it gives you the option of putting Illumina or nanopore, and obviously if you've got nanopore data, it's just one. If you've got Illumina data, it will be the two. And then what it does is it gives you, these are the list of things, explanations of how it does what it does and what it is describing. So how it describes sensitive, how it describes pre-MDR, XDR, other, for instance. Again, yeah, this is just explaining how it defines what the results will be. So what the substitutions are, what deletions are, and again, having a basic understanding of how this is phrased is probably helpful when you start to interpret it. But yes, the mutations are in reference to H37RV. So it only uses H37RV, and that's what it compares them to. So we can have a look at the reports, but actually I think what would probably be more fun is to look at it on TB Profiler. Yeah.
Yeah. So we did that we can put in fast Q and fast 5 files.
So I've very much got a here's one I made earlier in case we couldn't upload it in a sensible time. So I've already uploaded a file, and what it's
Did you upload the fast Q or the fast 5?
fast Q, you can't upload fast 5s.
Okay.
It won't work on fast 5s. So what I've done is I've already uploaded a file and it's given me this number. Keep hold of the number because it never gives it to you again, and then you've lost it. I find that very annoying. But what you can do, so you can upload it. Obviously, it might take a bit of time. It depends how many people are doing stuff on the server. Save that file number, and hopefully, there we go. It will bring up the results of, I can't even remember what this was, but it was some kind of TB. So this is the overall summary. So this is the very basic information. You can see that it says, this is lineage 2.2.2, and it's pre-XDR TB. You can also see with these green boxes, you can download this information as any of these types of files. So as a table, as a text file, a PDF, or the JSON file, which I don't recommend because it's a bit complicated. So then you get to the drug resistance. So these, if you can see, I'll just go all the way down. These are all the drugs that it checks resistance for, so quite a lot, and some of the newer ones. And this is what it's saying is that up here, these ones are what it's resistant to. And then the supporting mutations are basically where it's saying the mutations are. So this one is resistant to quite a lot of things, rifampicin, ethambutol, etc., etc. And those are the resistant, the SNPs that garner it resistant. You've then got lineages. So this one's a Beijing. The more depth you've got, and this is why it's really important to be confident about the amount of data you've got, the more information you've got, the further, the more sort of resolution you get. If you have a really small file with maybe, I don't know, a depth of 10, you might be able to say this is lineage 2, but the more information you've got, the further into this you can kind of go. So this is saying 2.2.2. So this was a good file because I got this off, we did this with some of the clinical trials. So we knew what this was, and we know that this is accurate. because we've done phenotypic analysis and also Illumina from it. It then goes a bit further into some of the resistance mutations. And it also, as I say, it keeps going with all of these mutations, how confident it is. And then it also gives you information about coverage. Sometimes it brings up a table from IGB, I forget which way around it is, that actually shows you this sort of the stack, but this one doesn't seem to have done that, so I'm not sure why. Anyway, so it gives an awful lot of information, but obviously you have to give it an awful lot of information for it to tell you what it is. So this is why having really good quality, high depth data is very important. Does anybody have any questions about that so far? Keith, you're probably the best person to actually fully explain what these results specifically say.
It's something like you have said.
Well, that excellent. Thank you. So yeah, so it's very, very simple to do. You upload it, it gives you a number. I think if you ask it, it will give you an email saying it's done, but I would just keep the number in like a spreadsheet or something, and then you can go back and it will bring this up. I did this, I want to say a few months ago, this specific run, and it's still there, but I know that they delete them after so long. So it's definitely worth saving it as a PDF or a text file or whatever you want, because I think they do delete them after some time, because I suspect you must have a massive server to keep all of this information on. Anyway, yes, so this is just kind of what we've gone through. The next slide, if you save it as a JSON file, looks like this. And as you can see, it's a lot less simple to read. So it gives you lineages. So this is a different one. You can see this is lineage 4, and it's gone down to 4.1.2.1. And it also does give resistances, but it's a lot more difficult to see exactly what it's telling you. So what I would do is actually save it as a text file, because this looks much easier to read, in my opinion. As I say, it gives you the lineages quite nicely, and then it literally tells you what it's resistant to. It looks a lot more like the output from the browser version. So yeah, if you have lots of them, there's also an option, although this also requires line command. If you've got lots of samples, you can create this nice chart. So it gives you phenotypic similarity, and it also gives you a table of what these things are resistant to. But yes, that might be a bit above this introduction to TB Profiler. So yeah, we do have in the drive, I think I gave you an example FASTQ file, and obviously you've made your FASTQ files, so you could upload those now to TB Profiler. Yeah.
Just wanted to add that because sometimes we talk about how drug resistance is spreading and how it is
Yeah.
treat and the drug resistances. You can actually see if you are having transmission of drug resistance, because you'll see that they will cluster together, and then they'll also have a similar drug resistance profile. So it's a very good tool. It's a snapshot of what might be happening in your area.
Yeah. The basic tool that doesn't require line command gives you that output, but once you get more confident with line commands, so obviously you're going to practice now you're the experts, you can then get onto the point where you can start producing outputs like this. This is not the only way of doing it. There are other ways of doing it, but this provides this kind of nice tree, but also resistances. And like Keith said, it gives you a bit of a human eye version of what is or might be happening. So yeah, as I say, you've now got FASTQ files. So bear in mind that the FASTQ files that you've just done are one single file. What we can do is concatenate them together. So I can show you how to do that, I think, on this computer if I've got some. Or you can just upload the single file itself and just see what happens. What you'll probably find is, because it's one file, you won't get as much information as this one. But let me just see. My laptop's doing quite a lot of things at once, and I'm a bit worried about overloading it. Where should I go? Might have to just re-download the FASTQ files because I think I deleted them all. Oh, that's what I want. I want the concatenated. Oh no, I don't want that one. I want all of them. Right. So you should hopefully have all of your FASTQ files in the same folder, I'm hoping. So, oh, where's that gone? No, I'm on the wrong thing. Right, that's what I want. So, yeah. So you need to bring up your, oh, go away. So you've got this, so this is Windows. John can perhaps check because it's cat, isn't it, on Mac for concatenating? Yeah. So there's two, so it's the same concept, it's the same command, but they've got different names. So in Windows, you need to type, in fact. Let's check that. Yeah. So what we need to do is type because that's the command that we're asking the computer to do to combine all of those things. In fact, actually, the best way of doing it is to actually be in the folder that you want to be in the first place. So if you type CD, that means change directory, and that's telling your computer to move into this folder. Oh no, I've got to move it onto my C drive. This is why you should always use the C drive, because my computer is networked to my university, and that makes life very tricky. Let me put it on there. Come on. Yes. It can't go. Come on. Oh, come on. Right, okay. Wait. Oh, come on. Yeah, so you should be, so what you will have got is all of those files that you what you've done is you've base called the files, but there's so when you start a sequencing run, you can, there we go, so you can ask it to put your files in a certain size. So as the default, it sets them at 4,000 bases, but obviously if you've got millions and millions of them, you're going to have lots and lots, or 4,000 reads, sorry, it's going to be a lot more than that. So what you need to do to get one file for each of your isolates is to concatenate them, which if my computer can cope, I will show you. But as I said, you need to have it on your actual C drive, because if you have it on your, as I say, your network drive, it doesn't like it. Right, so there we are. So what I'm going to do is copy the address. So CD, as I say, means change directory. So you are telling your computer you want it to go into C drive, data, FASTQ, and what you'll see is that it will actually change the original. You can see it says C users, and that's my username. It will now change it, and that's now, it's, the computer is now looking in that folder. If you weren't in that folder, you could then add the the address, but it's just easier because this is doing one thing in one folder just to move into that folder. So then what we're doing, as I was saying earlier, is add the command. So I don't know why it's called type. It's a bizarre thing to call it. It makes more sense to be called cat, which it is in Mac and Linux, but in Windows it's type. Then what we need to do is tell it we want to concatenate all of these files. So if you type in star, what that means is I want you to do this to any folder that has the ending fastq. So what's that saying is all of these 3 files will be concatenated into the one file that I am now saying to call this as an output. So this little arrow is saying, right, now move all of those into this file, which I will call cat1, for instance. And that's got to be, yeah, you could also, for instance, gzip it, so make it smaller, but just for now, let's just do it as that. And that, you should see, if this works, in this folder, it will come up with a file called cat1, which I've just made, and it should for goodness sake, it should in theory, why are you being difficult? It's done it for you. Okay, well, at least somebody's managed. Yeah. I don't know why my access is denied. Why does this keep happening? I don't know. Maybe it doesn't like that I put it in data. Let me try again and put it somewhere else. Let me put them in there, and that might help. I don't know why it's now saying everything has to be done with admin rights, but basically you should see, as long as you have admin rights to use your own laptop, that you will get that file that you've said output. Oh, for goodness sake. Right, let me try again. So CD into here. So now I'm in there. So now type, and then all the fast queues, and I want to call it cat1 fast queue. And we need a space. So what we should see once I run that is that that will hopefully appear here. There we go. Ta-da! So what you should see, if I open this up a bit more, is that cat1 is the size of all of these 3 combined, because that is literally what it's doing. It's gone along, it's found the first one, and it's adding all of the second one to the end of the first one. So if we opened it up, which you can actually have a look at, if you open them all up, and you can read them with Notepad, you'll see that here's my fast queue files, and what this is just all 3 stuck together. So the ones in the second and third one will be down at the end, but as you can see, it's a massive file, and also it goes that way too, so there's a lot of information. So what you can now do, as I say, is you can upload that cat concatenated file to TB Profiler. So
TB Profiler generates
yeah, TB Profiler. No, you have to make the consensus file. Yeah. Oh, sorry. Yeah. So you just press upload, and you drag your files in. You tell it that you want nanopore, and you want single reads, and then you pretty much just press submit. So if I leave the original address up, you can see it's not particularly long. It's tbdr.lshdm.ac.uk. Let me also leave up that command as well. If you're doing this on a Mac, it's the same. It's CD and then into your file. But then instead of type, you just put cat. It's the rest of it is still the same, but you just put cat instead of type. What's on?
Oh, it's great to get you to know that. Welcome.
We might find that if we're all trying to upload these files together, you might find it, it starts to struggle, but we can still give it a go.
Yeah, the files can be listed like that. Yes, okay. Space.
That's fine. You want some, yeah. So we'll give you that number, let's say it was on there, they're the same, we can still find out anything else. So if you say that number. It then does it. And then if you come back to it, I mean, try and add it now.
You said not to compartment that. Yes.
So if you go back to
All
the-
the yes,
and then type that number in there. You might say, probably say, I haven't done it yet. Eventually, once we've done it, we'll get that. So that's got the hero. Right, let me come around that side. Right, so how far have you managed to get? What's that? Right, so it's now zero in time, so that's
Oh,
what's it.
And I put a pen.
So not in class 5. Absolutely, it's fine. You look. Maybe the end of your mark, you're one. Which is just, oh, well, yeah. So it's not above class 5.
So that's the name of the file.
Class 5, all the way back to 5 within the folder. But you have to put class 5 at the end there.
Doing, they get lost somehow, sometime.
Yes, it's so old, sometimes we just.
Oh, yeah.
Now we can say, oh, this is 5, that's where you're right. Right, on the right folder. Oh, you can put the links where you were to go. So again, it was everything you need. That's how put it in the stack folder. So we'll put it in the folder of it. You have to click the folder on the right field again. You have to give it to all of them on this. So that will do that.
You can just look.
And it's really weird. Yeah, well, it's how to do something. So, so yeah. There we go. Yeah. And you found, where is it? Oh yeah, there you go. Yeah, there you go. See,
That's why you need to, if you use them that one. Extra. So when you get them, these
It's not very easy. So you can check, you can just list it and see your top one. Yeah. And you can check that business.
That was also just the so I found, and then it's like really good. So I found it, say, 14, if it's the other way around, depends. So I tend to use because it's easier to see and understand it.
You can go to report that.
Yeah, I use it. Yeah, but no, it's really nice. Yeah, again, it's nice.
Which was very.
They really do keep up on it, I think.
So are you able to then download the message that you went through to the profile?
So if you do it, so that is the advantage of doing it. I don't think you can download it on this, but if you do it by line command, you can see an output. It's
Yeah.
So it gives you, don't know what here, I think.
Oh.
Or will I have deleted it all because I tried to make space? I think I might have deleted it all. But what it'll give you is a folder called results, and then it will give you like BAM and JSON, and depending on what you ask it to output, it will give you all of those parts. So yes, that is one of the advantages of doing the online, the line command one, is to get those intermediate versions, which is what's really useful. It's good. It's going to. I'm impressed with how quick a lot of these pieces have been. It's a lot of data that they're processing, and they're not designed for this. It's impressive. To say, I was so impressed with my little laptop managed. It's really not coping with it. Has everybody managed to concatenate happily on their versions? Anybody got any results yet? To be provided? Might be slow because we're all now uploading stuff. We start recording this now.